Alrighty, Anna here. Let's go ahead now and talk about the spinal nerves and their receptors and neurotransmitters. And cross off neurotransmitters because we just went over receptors. Okay, so I'm going to end the recording and you're good to go. Before we go on, let's just make sure that we are all remembering the same thing. Okay, so the nerves are a bundle of axons. Okay, everybody should remember that. But also nerves tend to be what we call mixed nerves, which means they are going to have sensory and motor divisions. Okay, their motors will be both somatic and autonomic. And the sensory will be both somatic and autonomic. Okay, so there's just going to be a mix of everything. All right, now mixed nerves can include or do include both cranial nerves and spinal nerves. You remember from other discussions that cranial nerves originate in the brain or brainstem, and spinal nerves originate off of the spinal cord. Now, motor neurons tend to have their neuron cell bodies located in the brain or spinal cord. The exceptions are some of the autonomic, okay? But sensory neurons that are going out into the peripheral nervous system tend to put those cell bodies in what we call ganglia, okay? And after we look at the next picture, we're going to go into more depth on the ganglia. So right here, all this picture is doing is summarizing the information. I'm sure we've used this picture before. So you've got your dorsal root, your dorsal horn. You can see the two sensory neurons coming in. This is where they synapse. Now, what I like about this picture is the way they've used color. So we've got in darker blue, somatic sensory, in bright green, autonomic sensory. We have in red, somatic motor, and we have in yellow, autonomic motor. On this side, they show the cell bodies. And on this side, they show the regions of the horns. So you can see right here, the somatic motor done in red, and the somatic motor cell bodies right there in red. The autonomic or visceral motor is there, and the autonomic visceral motor is there. Here is the visceral or autonomic sensory, there and there. And then this is the somatic sensory here and here, okay? All right, let's go ahead and talk about the ganglia. All right, so remember, the body does protect most of our motor neurons by putting their cell bodies into the spinal cord, or keeping the cell bodies in the spinal cord or the brain, okay? But if you are in the peripheral nervous system, you're going to use a ganglion. And so we can think of this as kind of like a little bag or sac in the peripheral nervous system, and it's going to hold cell bodies. It can hold motor ones for the autonomic nervous system. Most of them we tend to associate with the sensory parts, but it can hold motor. All right, the most famous ganglion that we've talked about, everybody should be like so sick of this ganglion, all right, is the dorsal root ganglion. So ganglion is singular, ganglia is plural, okay? And in the dorsal root ganglion, because it is the dorsal root, it is only sensory. The ones out in the peripheral nervous system that don't get special names always can be motor or sensory. But when it is motor, it is typically autonomic motor. We are keeping the voluntary motor in the brain and spinal cord. Okay? And do more of that later. Okay, let's actually look at the structure of a ganglion. All right, so here we are with our ganglion. Here is your spinal nerve splitting into the dorsal root and the ventral root. Here is the dorsal root ganglion. And what we're going to do is we're going to take this and we're going to blow it up so that this is the dorsal root, this is the anterior root, and you can see the swelling of the dorsal root ganglion right there. Okay? And then this is the spinal nerve. So remember, the spinal nerve is a mix of sensory and motor. So you can see that these are going that direction and these are going in this direction for the sensory. So ascending and descending. Okay. Now look at all these things that look like SpaghettiOs. These are the cell bodies that are all packed in. And what I like is you can see like right here, all right, it coming up, and then the rest of it will go out in that direction. And right here, you see one, it's gone up, and then it's gone out. So remember, these are those unipolar sensory neurons. And they're all just packed in there, super nice. All right. Let's go ahead and talk next about sensory receptors. All right. You can classify them based on the stimulus or their structure or their location. We're first going to talk about the stimulus detection. All right. So what is the stimulus type? Okay. We've got major categories. You have mechanoreceptors. And I want you to focus on this part of the word, mechanical. This is talking about the shape of the cell, all right? If you change the shape of the cell, it will pick up a signal, okay? So we're going to change the shape by pushing on it or tearing it. So these will pick up things like touch, pressure, vibration, stretch, those kinds of things. So it's all about mechanical shape change, okay? The next one is the thermoreceptor, okay? So I want you to look at this part, therm. That sounds like thermometer. Thermometers pick up temperature. So thermoreceptors are detecting changes in temperature, so cold or hot. All right, another stimulus type receptor is the photoreceptor. So I want you to think of photography, picture. All right, what do those things all pick up? They all pick up light energy. Okay? And then you've got chemoreceptors. Okay, and chemical. So these are going to detect changes in chemical. So we're going to use this for things like smell, taste, also all of your blood chemistry. Okay, so like things for pH or oxygen levels, all right, things like that. And then we have this final category called the nociceptor. So I want you to think of this part of the word, noxious. If something is noxious, it's unpleasant. So the noxious receptors or nociceptors pick up pain stimulus. Okay. All right, let's look at, actually let's look at location next. So the stimulus, or actually not the stimulus, we can also divide up, we can also classify sensory receptors based on the location of the receptor. Okay, and there are basically three broad categories, okay, which actually kind of subcategories, okay. You have the exteroceptors, and you have interoceptors. All right, and these can also be called visceroceptors. Okay, there's one more actual location category, and it's kind of an interoceptor, but then it's also kind of not. So we're just going to call it a different thing, and that is the proprioceptors. Okay, so obviously exteroceptors are all about stimulus outside the body, so things that are hitting the surface of your body. Okay, these tend to also fall into the category of either mechanoreceptors, all right, and or some of the special senses, so like photoreceptors, all right, some of the chemoreceptors, all right, those kinds of things. Interoceptors, so focusing on this intero or visceral, all right, these are going to be detecting stimulus inside the body, okay? These will often be things like chemoreceptors, all right, or thermoreceptors, or mechanoreceptors. I'm hoping everybody's starting to pick up on the fact that I'm merging these words. Okay, so I'm mixing them up to get better explanations of what the receptor does. Okay, so you can mix together the stimulus type with the location of the receptor. So I can have mechanoreceptors that are exteroceptors, and I can have mechanoreceptors that are interoceptors, and I can have mechanoreceptors that are proprioceptors. So what is special about the proprioceptors? They are specifically found in skeletal muscle and the joint structures, so tendons, ligaments, that kind of stuff. And they are very specific in that they monitor the position of these joints and muscles and degree of stretch. All right, so they basically are saying how contracted is this and how stretched is it. Okay, let's actually look at a picture of some of these before we go on to looking at stimulus by structure. All right, so this little picture I like because they start merging concepts. So L is location, S is stimulus type. So here we have a free nerve ending, all right, which could be an exteroceptor or an interoceptor or a proprioceptor. It depends on where it's at. So if it's in your skin, it's also an exteroceptor. If it's in your stomach, it's an interoceptor. And if it's in your joints, it's also a proprioceptor. Okay? Now, we can also take things and we can talk about them in terms of stimulus type. So if I'm looking for a pain nerve ending, it could be one that responds to temperature change, and so is a thermoreceptor, or chemical change, so it's a chemoreceptor, or pressure, and it's a mechanoreceptor, or just flat out pain, and then it's a nociceptor. Okay? So you can see you can do these broad categories. And so here's another way. Your exteroceptors, they tend to be mechanoreceptors with some exceptions, okay? So the Merkel cell would be an example of an exteroceptor that is a mechanoreceptor, okay? And over here is just kind of organizing some of this information and redoing what I did on the blackboard just a minute ago. All right, so we've looked at classifying sensory receptors based on location and based on stimulus type, but we can also talk about them in terms of structural complexity. So we can break this up into two broad categories, simple and complex. I bet that was a huge surprise, wasn't it? Okay. So simple receptors are where we are just going to modify a nerve ending. Okay, we're just going to modify, let's say that we can even be the dendritic part of a neuron. Okay, and it's going to pick up general sensory information. This is the vast majority of our sensory receptors. It's just these modified dendritic endings. Okay, and they can be broken up into two categories. You've got first free dendritic endings, all right, and they're just basically a dendrite that's kind of floating in space, all right. And then you can have encapsulated dendritic endings. So this would be like your Merkel cell, where it's basically got a capsule that goes around it. So this would be like the little dendrite, and this would be like several little dendrites with a little cap around it, okay? And then you've got the complex. The complex, we're going to associate those with just the special senses. So that's the other thing with the simples. Those tend to be general senses. So the special senses are going to be just vision, hearing, balance, taste, and smell. Okay? Let's see, what else do I want to say about some of these? The free dendritic endings, let's see, we can talk about those a little bit more. Those are mostly going to be unmyelinated C fibers. And we've talked about those before. So these signals, since they're unmyelinated, are going to be slow. Okay? Slow. A lot of these tend to be mechanoreceptors. Okay, so a lot of mechanoreceptors. But also quite a few of our thermoreceptors. And also our nociceptors. So you've ever noticed how you might touch a hot pan and you don't immediately realize how hot it is? So it's like you like tap it, and then it takes like a second or two for your brain to tell you whether it was hot or not hot. So I do this all the time when I'm trying to decide if my pan's hot enough to put something in. So very, very slow. I mean, very slow. Okay. For these free dendritic endings, oops, I made a mistake up here. Merkel cell is not an example of encapsulated. Okay. So some examples of the free dendritic endings are the Merkel cells and the root hair plexus and various itch receptors, which are actually chemoreceptors. So I guess we can put that up here. Now, I know I got this mixed up. The encapsulated dendritic endings, examples of those would be the Meissner's, not the Merkel's, the Meissner's corpuscle, the Pacinian corpuscle, Ruffini's corpuscle. We've talked about the muscle spindles at some point, the Golgi tendon organ. These all have special little caps that surround it. Golgi tendon organ. Okay. And these are both proprioceptors. Okay. And again, what you have is you have the dendritic ending here, and then you've got some kind of capsule that surrounds it and protects the end of it. Okay? And I think that's it for what I want to talk about on these different receptors. And we didn't talk about neurotransmitters in this. I think that's in a different slideshow. So I guess I should go back to the very beginning.